Okay, so I'm going to start by introducing today's speaker. So today's speaker is Professor Clarice Aiello, and she is a quantum engineer interested in how quantum physics informs biology at the nanoscale. She's an expert on nanosensors harnessing room temperature quantum effects in noisy environments. Aiello received her PhD from MIT in electrical engineering and held a postdoctoral appointment in bioengineering at Stanford and then later at the chemistry faculty of Berkeley. She joined UCLA in 2019, where she leads the Quantum Biology Tech Qubit Lab. And I've just also a few minutes learned that Clarice also has a connection to Cambridge, where she did an MPhil degree a couple of years ago. So I'm extremely happy to have Clarice here. Quantum biology is obviously a topic that is increasing in popularity exponentially at this time. So super excited, and thank you so much for waking up at 3 a.m. Over in California.
Cool. Thank you all. I'm just trying to. Where am I? Slideshow. There it goes. Good morning, everyone, and thanks so much for the invitation. Again, it's 3 a.m. here, so I have my big cup of coffee. Cheers. And yes, uh, and it was very kind of you to say I got my MPhil a couple of years ago. It wasn't a couple of years ago. It was a long time ago. I'm getting old. So, but yes, I still cherish my memories from my Cambridge times and yeah. And so again, thanks for joining. And yes, I like to call myself a quantum engineer, right? Um, this means that I build apparatuses to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So let me try to explain the weird title of my talk. So as a quantum engineer, a quantum mechanic, I do know a little bit about spin physics. What do I want with biosensing, right? And I have to admit that beyond high school, I never took a uh, biology course. So where do I want to go? So before I explain where I'm going, I really need to tell you where I come from. So in the beginning of the talk, we're going to start talking about things that are probably very familiar to you about hardcore quantum mechanics. And by the end of the hour, we will have talked about things of biological relevance, such as organismal migration, how our bodies respond to radiation and to oxidative stress. And I hope I will have had time to talk a little bit how I think that we can learn with nature to build better technologies. And that's because as I was introduced, I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. So to motivate my uh, talk, I'm going to open a parenthesis. At some point, this parenthesis will be closed, and that when I close this parenthesis is going to be the end, the first break that I'm going to do, and that's mostly about my past. So I think that overall, humankind is obsessed with measuring things better because it sort of means or it implies that we might understand nature better. So we might think about measuring better frequencies, for example, with this atomic clock that sits at NIST, the National Institute of Standards and Technologies here at the US, and that helps us define super well what the second is using atomic transitions. We can think about measuring better magnetic fields so that the image of our baby is better resolved. And yes, this is a magnetic resonance image of a baby inside the mother's belly. And we can also think about measuring better accelerations so that our gaming experiences are increased. And those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this sense, you need, in this case, you need a tiny little sensor that can measure tiny little things. if the sensor is tiny, let's make it very tiny. Let's make it quantum. And in fact, there's a whole body of mathematical arguments that can prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. I'm going to give you one example of a quantum sensor that I played with in my past. In my past, I put a single spin in diamond, in the material diamond, to work as a quantum enhanced magnetic field sensor. So, how might that happen? How might we do this? Right. So, the single spin in diamond that I played with, it arose from a crystalline defect in diamond. This crystalline defect, I'll tell you its name in a moment. This crystalline defect is a color center. Color centers in diamond are the crystalline defects responsible for the color of those nice diamonds that you see there. And color centers have the property that they absorb light and then they de-excite and they fluoresce. And that's exactly why you see the color of those diamonds there. Again, the defect that I work with, and it's sort of a hyped defect now, it's called the nitrogen vacancy center in diamond. It occurs naturally, but it can also be engineered. And it happens when a vacancy, a missing carbon atom, sits next to a nitrogen atom, which is the most commonly occurring substitutional impurity in the carbon lattice. When those two things happen together, when the nitrogen sits close to the vacancy, there's sort of a mess of things that happen, right? There are the unpaired electrons of the nearby carbons. There might be extra charges floating around, but something extremely nice happens when you start calculating the quantum mechanical energy levels of this mess. When you calculate the quantum energy levels of this mess, the picture simplifies considerably because those energy levels really look like the energy levels of a single spin. For the experts, it's actually a single spin one, okay? But we can single out two of these magnetic sublevel states out of the three and make a pseudo qubit, pseudo spin out of those levels. But that's just for the experts. So, and the way that we look for those defects in the lab at room temperature is the following. We place a diamond slab. It can be a crappy diamond slab. In fact, the very first diamond that I worked with was given to us as a discard by a jeweler, right? So you put the diamond slab on top of a moving stage and you move it around at the same time that you shine laser light of a particular frequency onto this diamond sample. Actually, it doesn't need to be laser light, but it's more convenient and more efficient if we use laser light. So we're going to, to try to excite this centers that are below the laser path as we're scanning. And the hope is that the moment that we hit one of those centers, they will get excited. And as they de-excite, emitting fluorescence, we can sort of collect this fluorescence to image those defects. And this is what it looks like at increasing zooms. Okay, so at this, I also have always have to think, as you see here in this bottom right most picture here is in this square of about 1 micron sized is a blob, a single blob, okay, which is actually quite remarkable because this blob is the signature of the fluorescence of one single spin. Again, the defect itself is atomically sized, but this blob in this case is diffraction limited by the optics that we use to employ that. But even though this blob is big, it's super cool because we are getting information uh, about a tiny microscopic uh, effective single spin via its fluorescence. Compare that, for instance, with nuclear magnetic resonance, right? You would never be able to get information on a single spin using nuclear magnetic resonance methods. And here you can. And in fact, this effect is even more special than that, in that it has, and that's jargon, a quantum state dependent fluorescence intensity. This means that just by looking at how strongly this blob is emitting light, we can infer if the spin was up or down. And this is going to be used to measure things as a very good experimental handle that we can have on this defect so that we can read out the state of the spin. And actually, I'm going to come back to this property later in the second part of my talk, but the NV center has a quantum state dependent fluorescence intensity. But I promised you that it could be put to use as a very sensitive magnetometer. So here's how it works. Um, Here I write two states of this effective pseudo spin 1/2 that I call 0 and 1, which just represent two different energy levels of this property of this spin states. Right. In particular, 0 and 1 are separated in energy by about 3 gigahertz on like in the microwave range. And it turns out that one of those states, the state marked here is as 0, is insensitive to external magnetic fields, whereas state 1 is sensitive to external magnetic fields. So that in the presence of an external magnetic field, state 0 won't care, and state 1 will be promoted to another energy level, if you remember, by something called the Zeeman energy. And again, this tiny little delta in energy that the state 1 suffers is proportional to the intensity of the external magnetic field. And now, this point doesn't get made so often, but I really think it's important in that the problem of measuring magnetic fields with the spin is effectively mapped onto the problem of being able to measure a little detuning delta from a known resonance. So uh, we the spin in diamond is going to give information to the experimentalist on external magnetic field strengths just because the experimentalist knows how to measure the tunings from resonance. And in fact, experimentalists and electrical, like physicists and electrical engineers, have known how to measure the tunings from resonance for decades now. And that's all the reason why this works. Uh, but again, this method of getting magnetic field information only lasts while the spin is well described by the laws of quantum mechanics. I don't think it comes as a surprise to anyone in this audience that everything that starts quantum dies classical, and the statistical time that it takes for something to be well described by quantumness to go into something that can be described by classical physics. In this sense, the time that it takes for a spin to have a well-defined phase relationship between up and down until the time that it takes for the spin to be considered as a classical magnet is usually referred to as coherence times. And in the case of this defect in diamond, the coherence times, like naturally occurring coherence times for the experts T2 star, is about 2 microseconds at room temperature and in the messiness of the solid state lattice. And actually people now it's a whole materials problem now to make diamonds that can increase coherence times of those defects. But naturally occurring crappy diamonds already have a T2 star of 2 microseconds, which is, for the people who do things at room temperature and in quantum information, 2 microseconds is usually huge. I have to convince you that 2 microseconds is very, very large. But the whole field of so-called quantum sensing or even quantum information, for this whole field, the name of the game then is like, can we play with the spin can we control the spin state so that uh, we can play with it for longer as a quantum sensor. And as I am sure that many of you have heard about, and this has started in the field of nuclear magnetic resonance like half a century ago, the way that experimentalists do this control to extend the quantumness of a quantum system is with applying engineered electromagnetic fields onto the quantum systems of interest, right? So the idea is that, uh, very put very simply, uh, you can only acquire interesting signal from this spin for as long as it's quantum. So if you can kick it with electromagnetic field so that it's quantum for longer, the longer you can acquire a signal for, as you Fourier transform the signal, you have a better resolution in the frequency domain. So, which is going to give us a better resolution in whatever you want to measure, in this case, magnetic fields. Okay. So, before I give you one instance of how we do this control in the lab, I just want to point out that don't forget the big picture here. The big picture is to have a tiny little sensor that can come very close to a tiny little sample and that can measure tiny magnetic fields produced by the sample, because this spin reacts in its energy levels to tiny magnetic fields, and the experimentalist can measure this. So never forget this big picture. And now I'm probably going to go to the most technical, it's not going to get worse than this, the most technical slide of my talk on how we can do one experiment of controlling the spin to extend its quantumness. Okay? So this is just an experiment, uh, a way that we work in the lab to extend the quantumness of this sensor. So I told you that experimentalists like to apply tailored electromagnetic radiation uh, to, to kick this spin in very particular times. In my past, one of these sequences of kicked electromagnetic pulses that I worked with is called rotary echo. And what this rotary echo sequence of pulses mean is explained in this graphic there to the right in nuclear magnetic resonance language. Let me decipher this for you. This means that I am applying CW radiation onto the resonance frequency of my spin, but at predetermined intervals, I am going to flip the phase of this microwave pulse, okay, while keeping the excitation going. If you will, if you're familiar with this, it's just a uh, Rabi experiment where the pulse phase that you're applying is flipped at particular times. This pulse sequence, this rotary echo, has nuclear magnetic resonance origins, and it's known to correct for imperfections of the microwave that you apply onto your two-level system, onto your spin, onto your qubit. This sequence of pulses is also known not to correct for detunings from the resonance, which is a good thing because in my previous slide, I uh, sort of argued that the tunings from resonance is what are going to give us information on the magnetic field. And I won't have time to talk about this, but like the length of the time that is spent before you do this phase flip actually can help correct for different types of noise that your qubit is uh, subject to. Okay, and so it's this rotary echo is actually a very nice pulse sequence to do magnetometry with uh, single spins, for example, in diamond. So if you apply this CW with phase flips microwaves onto your spin qubit, this is sort of the signal that you get. Signal here is just the normalized um, intensity of light that this spin is giving back at us. So the way you can read this is like at the top of the signal at values close to 1, your spin is, say, in state 1. At the very bottom of the signal, your state is in your qubit is in state 0, and for everything, every value in between, your spin is in a coherent superposition of 0 and 1. So this is sort of the signal that you get. We can apply a formalism known as average Hamiltonian theory to get to first order, like what sort of information the signal is giving to us. And this sort of formula was not known before. We spent a lot of time trying to come to, to this formula. And now we know how the signal depends on the uh, uh, detuning delta that we want to measure. And again, this detuning is going to give us information about the magnetic field. And we can, again, with the help of this formula, and we can Fourier transform this signal to look at this as a spectrum, okay? I won't have time to explain this, but um, as you extend your time during which you apply this field onto your spin, you can get better and better frequency resolution. You can get better and better information about those tiny little delta detunings, and the smallest detuning here that this signal actually is giving information to us about magnetic fields on the order of 1/10 of the magnetic field of the Earth, which is tiny. We're going to talk about how tiny the magnetic field of the Earth is in a moment. But with this spin, in this particular instance of our experiment, we can already use the nitrogen vacancy center in diamond to get information about magnetic fields that are about 5 micro Tesla. So again, that was just one example. The big picture is this desire to have a little sensor that can come in very close proximity to a sample, and therefore can give us information very localized on like magnetic fields in the sample. With this, I'm going to make the first pause for questions. Okay, and but before that, and while you think about your questions, right, I just want to make sure that that you agree with me that I've just presented to you a quantum sensor that works at room temperature and in noisy environments. So if you have your question, ask it now. If you don't have a question, what I want you to do is to Google what thermal energies are at room temperature in gigahertz. So I've just told you that this spinning diamond has a different a resonance at about 3 gigahertz. And if I were you, I would Google right now what KBT is in gigahertz, and tell me if you're astonished by that or not. And please ask me whatever we want about how KBT compares with this resonance of the qubit right now as well. So with that, I'll shut up and ask for questions.
Okay, thanks, Clarice. I'm desperately trying to Google this question now. Uh, I'll get back to that. I can't see any questions in the chat. Okay, we have a question from Thierry
Oh,
Ferris. I'm going to
yes,
bring Thierry Ferris to the panel then, so Thierry can ask the question.
yes. Hello there.
Thierry, you're muted, Thierry.
Sorry, my computer is a bit slow. Hi, Clarice. Thanks for the first part. Actually, I have a funny question.
Yes,
So you mentioned this corresponds to 3 gigahertz,
yes.
but now we all know that the range of mobile phones span from 1.6 to 3 gigahertz each. So does it mean I can operate that spin with my mobile phone, Nelly, or disturb it?
Probably not, because I would hope, and I really, really hope that the signal emitted by your phone is not that strong,
I
right?
mean, it's a fair question. I mean, okay, maybe I
Yes, yes, yes,
presented
yes, yes. No, no,
it
no, no,
a
no, no,
funny way, but
no, 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 no. Yes, you absolutely could, but the Rabi frequencies that you would get would be extremely small, right? So the way that we sort of excite this uh, center with microwaves is that you have your confocal microscope, you have like your objective,
yeah.
right, and you place the tiny slab of diamond below the objective,
Mhm.
and you sort of run on top of your sample well, nowadays there are people doing like microscopically lithographic like the microwave like chips, but the crappiest and easiest way that you can excite it is by pushing a tiny little 25 microns wire of like a tiny little wire on top of the sample, right? And relying on the proximity and push through a microwave, a very much amplified microwave at 3 gigahertz through this wire. And then you rely on the magnetic field of this current to, to excite, to give this RF. So uh, this defect needs to see a lot of needs to see strong microwaves close to it, right? So and this microwave is really really super amplified, right?
Yeah,
So
I mean,
it's yeah,
the other question. I mean,
yes,
it's the reason behind that question was also because so we've been working in the lab with similar devices, NV centers,
yes,
but also on silicon, and I know that once you have defect, sometimes you may think uh, that the effect of, for example, microwaves coming from mobile phones are negligible because, for example, your cryostat is green because it's the amplitude is supposedly small.
right. And
But
you see it, right?
oh yeah, oh yes, I do.
This is so
Um,
cool. Okay,
so I mean, not on a positive way because obviously mobile phone they emit a bit
right,
they're not supposed to control qubits, so they mostly contribute to the noise background by exciting
right.
other traps. So
This is
typically
so cool.
your noise go from 1 to a factor 10. So if your signal is very small, then you lose it.
Oh my God, this is so exciting. It's really the first time that I heard
That's
this.
why I'm very harsh on the people bringing a mobile phone in the lab. There is a good reason. But
Oh my God, cool.
nowadays, even in computers, I mean the clock is in the gigahertz range. So except if you have a computer or laptop which is somehow screened, this could also be an issue. So it's a
This
big
is
problem.
crazy. This is crazy. So can you build a Faraday cage around the clock?
Well, you can.
Okay.
But you know, I mean, you have to put holes into it.
Yeah.
So you need to have either optics or cables. So you will have to also do filtering
Right.
because once you put a too big hole into a screen room or a Faraday cage with a big hole, then you're not doing much.
But that's
This is going
it's
to, yeah.
but that's super exciting information, and I'm sure there is an undergraduate project there somewhere or something like characterize the noise from mobile phones on keyboards. That's
Well, I
super
mean,
super cool.
yeah, I mean, I don't want to
That's
extend too
great.
much on that, but one experience was actually I could detect if somebody had a mobile phone in the room.
This is so cool.
So this already happened to me. I say, well, you have a mobile phone? No, no, no. My mobile phone is off. I say, no, it's on. And then actually it was on.
This is so cool. This is so cool. Nice.
Well, it's
Nice.
not so cool, but
No,
anyway.
no. Sorry.
Okay.
Yes.
So I think we have another question from Mercedes, and I'm a little bit worried with time.
No, that's fine. Thanks.
So I think we're moving on to Mercedes. If you would like to ask your question.
Hi, mine really quick. Yeah, probably simple. I was wondering, how were you able to determine which qubit was your reference qubit? Like which qubit you're going to use for measurement from the other qubits in the spin bath? And is it different at room temperature to determine the one that's the central qubit for measurement than it is at thermal equilibrium?
So the only qubit here, proper qubit, in this what I presented, is your magnetometer, right? Everything else is, in principle, it can be treated not as a qubit, but as a magnetic field source. Okay, so the only qubit that you care about is the nitrogen vacancy center qubits, and say nuclear spins in the sample, right, that could be characterized as qubits. You can forget that they're qubits. You can treat them as sources of magnetic field that move the energy levels of your qubit of interest, right? So in fact, given any position of your electron spin tip, there might be several nearby, say, nuclear spins or other electron spins contributing to an effective magnetic field that you see.
Really good. And which one is your main source for the magnetic field in the magnetic field bath then?
So it depends, right? It depends on what exactly you want to measure or not. So there are sources of magnetic fields in the bath of the crystal itself. So carbon-13 nuclear spins, if you're dealing with a dirty diamond, nuclear spins, phosphorus, right? Spins are also sources of spurious magnetic fields because they're in the crystal of your where your sensor lies. But materials people are working to get rid of those spins in the diamond lattice, right? But then the spins that you might be measuring from the sample depends on the sample itself. And usually you have, right now, I think you can, I think people could very well, a couple of years ago, measure single hydrogen spins as a signal in a configuration like this, in a sample.
Thank you
like
very much.
nano MRI or something. But nice seeing you, Mercedes.
Yeah, it's all good. Thank you.
Okay, I think let's do one more question before we move on. We have a question from Shushi.
Oh, hi. Hi, thank you. So I want to know whether this pulse sequence could be applied to ensemble NV measurements where the inhomogeneous broadening is the dominating broadening.
So, I mean, all pulse sequences can be applied to whatever. So what the assumption here is that you would apply this sequence to an ensemble of NV centers for them to act as a
ensemble DC magnetic field sensor.
So if I understand correctly, the inhomogeneous broadening is a broadening in the deltas, right?
In strain splitting, yeah.
So the property of this sequence is that it does not correct for detunings from resonance. So my first guess at this point is that it wouldn't help with the inhomogeneous broadening.
Ah, right, yeah.
Right. On the other hand, if you have a crappy microwave source, this might help because this actually corrects for imperfections in the X excitation, not in the Z.
Yeah, so in this case, how does this compare to the other pulse sequences? Like,
Yes, great. That's a very important question. And basically, the best pulse sequence ever to sense DC magnetic fields is a pulse sequence known as a Ramsey sequence, right?
Yeah.
Uh, and it was also proven that there's in one extreme a Ramsey sequence, and on the other extreme, it has been shown that you can actually use a Rabi frequency, just a continuous microwave pulse, to sort of also measure DC magnetic fields. So the two extremes are then Ramsey and Rabi. Those two things are different in different environments. A Ramsey sequence, whereas it's the most sensitive in terms of how good your sensor is, if your the cohering noise is super strong, right? The time that that the spin is in a superposition state and made to evolve, like if your noise is extremely strong, you won't be able to acquire the signal for long.
Mm.
So on the other hand, the Rabi frequency, if you apply just a Rabi pulse, you actually get that your signal is proportional to a cos of delta squared. So the sensitivity is not super good, but kicked quantum systems, they live for longer. So if you are in a region of Beth with a very, very strong decohering noise, if you apply a continuous decoupling, like if you apply a continuous microwave, you can make your qubit live for longer with a trade-off in loss of sensitivity. So the way we like to think about the rotary echo is something in between, right? Because the longer you sort of take to flip your face, the closer you are to the Rabi experiment. And on the other hand, sort of we can show that by flipping sort of fast, you are very close to the Ramsey. You all actually get to sensitivities that are just a little bit worse than the Ramsey. And with the rotary echo, you can actually, in principle, the experimenter can change this pulse length to match whatever environment your sensor is currently immersed in. So we like to think we like to think of the rotary echo as a very flexible sequence that can correct for different types of noise depending on that particular experimental realization.
All right. So by cleverly choosing the frequency that you switch the phase, that will allow you to sample different part of the noise spectrum. Then.
Well, it's not sample different. Well, I think you could see that one could do like noise spectroscopy with this. Yes. So I'll take the way that you say this, but the way that we usually think about this is that we can adapt this pulse sequence to correct or to be resilient to the particular type of noise that is currently present in your experiment.
Right. Cool. Thank you.
Cool. So I'm going to start going forward, right? Because and now, before I close my parenthesis, you should have Googled that in gigahertz, KBT at room temperature is about, I think, 6,000 gigahertz or something. And one question that I don't usually get for NV but that I usually get from what I'm going to talk about now is what happens now. So your gigahertz, your qubit frequency is much smaller than KBT. So how can you use it as a quantum sensor at all, right? So the answer to this, and it's important that I mention this now, is the fact that yes, KBT is much larger than the qubit difference in energy, but all the quantum information that we're able to obtain with the sensor is only coming from times before the qubit comes in equilibrium, thermal equilibrium with the environment. The time that it takes for the qubit to come to equilibrium with thermal equilibrium to the environment is about, in this case, more or less without any control pulse, about 2 microseconds. Okay, so it is possible to obtain quantum information from sensors that have a transition frequency much smaller than kBt if the information is gotten while the thing is in a proper quantum state, is well described by the laws of quantum mechanics. Okay, and you're going to see why I wanted to mention this important thing right now. So now I'm going to the second part of my talk to talk about what I really want to talk about. So before in my life, those were the types of sensors that I was used to working with. But at some point I realized that, well, nature is a very good engineer, right? It has had millions of years to perfect their sensors. And in many, many cases, natural sensors overperform humankind made sensors in crazy, crazy ways. In my past, I've worked with two types of natural sensors, a light sensor that is not quantum at all, and a magnetic field sensor that I'm going to argue seems to be quantum. Okay, so let me talk to you about this natural quantum magnetic field sensor. But in order to do so, I'll have to start with biology at the nanoscale, namely the chemistry. So it's been known in basic chemistry that for decades now, that magnetic fields can alter the final products of a class of chemical reactions that involves spin correlated electrons. And yes, by spin correlated electrons, I mean that they are entangled. Okay, so let me tell you how this works in chemical reactions. Again, no doubt that this is what happens in the lab when chemists are playing with compounds. So imagine that. So, and now you should be looking at me. I have my props. I carry my spins around. So imagine that you have two spins at the outer shell of a, say, fluorescence protein, or like a chromophore, something that can be excited, and as it excites, it emits light, and this light is known as fluorescence. So those two spins, they are in a singlet state, one pointing up and one pointing down, just because either of the Pauli exclusion principle, or you can think about it in this because that's their most energetically favorable state. So if a photon comes in, it gives enough energy of appropriate frequency, it gives enough energy for one electron to be promoted, and most times this electron will come back down, emitting fluorescence. Sometimes though, upon absorption of a photon, this guy here will be excited, and before it excites back, sometimes it meets with another electron spin in solution, a free radical. Free radicals are very, very common to find in chemical reactions. Okay? If those two spins meet, okay, the first point that I want to make is that because photon absorption processes are spin conserving, whenever this guy meets with this guy, they need to start in a singlet state because those, in the absence of any other laws, because those two spins were in a singlet state. So I hope you agree with me that in the absence of laws, those two spins need to start in a singlet state. Well, singlet states, people sometimes we forget that singlet states are spin correlated, are entangled. So it's in this sense that this mechanism depends on entangled electrons. The second thing that you need to know is that there are chemical reactions that are spin dependent. This means that the chemical reactions, they continue through one branch if the spins are, and now it's jargon, measured by the environment to be in a singlet state, and the chemical reaction continues through another branch if the electron spins are measured by the environment statistically to be in a triplet state. Importantly, the final products of those two branches are macroscopically different. Okay, so a putative fast quantum mechanical phenomenon that happens at the top of a chemical reaction where electrons can be found either like this, or they can alternate. A fast process occurring at the top of a chemical reaction that changes spins can have macroscopic signatures downstream at much longer timescales. Okay. Third thing that you need to know is that in this case, those two electron spins are actually see different local magnetic field environments. So this electron here, it's usually coupled to nuclear spins with the molecule where it came from, whereas this electron spin here, it's free. So in the presence of an external magnetic field of a strength that is small compared to the hyperfine interactions, this guy will Larmor precess around this Larmor frequency of the external magnetic field, whereas this guy will Larmor precess around the frequency of this external magnetic field plus the Larmor frequency of this hyperfine interaction, which sort of dominates. Okay, so in the presence of a weak magnetic field, this guy is doing its thing. And if this external magnetic field shifts a little bit, this guy doesn't care, but the Larmor frequency of this guy is strongly altered. This means that actually, upon tiny changes of a weak external magnetic field, those two spins are going to pick different phases with respect to each other, all because the Larmor frequency of this guy changes by a lot. So the guy who's doing all the job in picking up, so that those two spins, as they Larmor precess, pick up different phases, this guy is the one doing all the job. This difference in phase actually translates into a difference in probability that the spins are measured by the environment to be in a singlet or in a triplet state. All I'm saying is that an external weak magnetic field or changes in it is going to alter the probability that the spins at the top of a chemical reaction are found in a triplet or singlet state. So effectively, an external weak magnetic field is going to alter chemical pathways in chemical reactions, can alter in specific molecules. Okay, so this is known in chemistry, and all the ways to explain this pass through a dependence of this process on the spins of involved. Okay, there is no alternative phenomena. There's no alternative explanation to what happens in chemical process. Okay? So, and now, even though this is not a bird talk, I need to start talking about birds because in the 70s, many years ago, biophysicists, theoretical biophysicists, they wanted to understand how birds actually are using the magnetic field of the Earth to migrate. There's no doubt that birds, when they go north to south, they use the magnetic field of the Earth at least as a partial cue. And again, the magnetic field of the Earth is about 50 micron Tesla, which is weaker than the magnetic field that you feel when you put your cell phone close to your head. So how might they be doing this? So the theoretical biophysicists, they proposed that were the same type of spin-dependent chemical reaction at work physiologically inside the bird somehow, right? Birds could sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from this sort of spin-dependent chemical reactions. And if that were the case, this would mean that there is a transient coherent process that is spin process that would be happening inside the bird. And at that point in time, the only and because this process that I just described usually starts with the absorption of a photon, right, that excites this guy, scientists, biophysicists started looking for molecules, biological molecules that absorb light. So the first thing that they looked at were photoreceptors. And at that time, the only known animal photoreceptors known to sustain, to be able to sustain such a spin-dependent chemical reaction was a photoreceptor that is present in our eyes, in the eyes of migrating birds, in the antennas of migrating butterflies. It's a protein, a photoreceptor called a cryptochrome. Cryptochrome is a protein that has a flavin. The flavin is the thing that makes it fluoresce. Again, it's a photoreceptor, but interestingly, it's present, and I think this statement is accurate. It's present in all our cells because it also has circadian rhythm regulation functions. And actually, and this is not weird science. The reason why when you're jet lagged, people say, "Oh, stare at blue light," is to sort of try to change a little bit the workings of your cryptochromes that absorb in the blue spectrum. Okay, and it turns out that beyond birds, there are many organisms that seem to be magnetosensing via ways that seem to depend on cryptochrome. First of all, cryptochrome is absolutely universal across the tree of life, from microbes to like humans, right? To plants, to reptiles, to birds. And so in this slide there, those are some organisms that express cryptochromes, and boxed in red are organisms that studies, there have been magnetosensing studies that have shown sometimes weak, but correlation of magnetosensing with cryptochrome. And yes, humans are right there. And actually, evidence for cryptochrome-based magnetosensing is really widespread, but unfortunately at disconnected length scales. Okay? In the very tiny length scales, and it's in the left part of my slides, there are chemical studies on cryptochrome in solution that sort of show that cryptochrome does work as a quantum sensor of magnetic fields at room temperature. Let me go through some of the evidence that you see there to your left. Okay, so in one study, researchers captured the fluorescence emitted by cryptochrome as a function of the time during which you excited cryptochrome with an appropriate frequency that could make spin transitions. Okay. The first thing that you see, this is for cryptochrome in solution. The first thing that you see is that this cryptochrome is emitting fluorescence, but this fluorescence is going down. This means that you're killing sort of your sample by applying very strong laser fields onto that. But as the researchers pulsed a tiny magnetic field on and off as they were capturing this fluorescence, and this is not an artifact. They have tried different magnetic field on and off frequencies, intensities. What they see is a tiny fluorescence modulation as the fluorescence is decaying, right? The fluorescence comes up and down. I'm going to argue that this is an analogy with the single spin in diamond. For the single spin in diamond, depending on how strongly it was emitting light, you could infer if it was up or down. Here, for this cryptochrome, just by looking at how strongly this guy is emitting light, this protein is emitting light, you can actually infer whether the chemical reaction continued through the singlet branch or through the triplet branch, or which combination of probabilities that was. Also, although this evidence is not super absolutely unquestionable, there's evidence that shows that seems to suggest that cryptochrome coherence times at room temperature are at most on the order of 1 microsecond. Again, which is on par with the 2 microseconds that I quoted for the envy center in diamond. Okay, so this is very encouraging. The problem is that the next step of evidence for this mechanism occurs at huge length scales. Okay, occurs for experiments with birds, experiments with flies, experiments with thousands of cells in solution. And those experiments, they are consistent with the things that people see in vials of cryptochrome, but it's really hard to say, well, the bird is behaving the way it is because there's some quantumness going on in it for a short time. So before I argue that we need to bridge those length scales, let me just describe some experiments done with say, birds. The problem with experiments with birds is that it's hard to make them. The sample size is small, but people do capture birds, migrating birds during migration season. They put them in cages with with shields, with Faraday cages around and coils inside. And by changing the magnetic fields that the birds see, the birds want to go out of the cage via different directions. People can also train flies. Okay, I don't know how they do this. It's harder than quantum mechanics to me. But they train flies to find food based on the presence of a magnetic field. Then the researchers knocked out, they removed the cryptochrome gene from the flies, and the flies were no longer able to find food based on magnetic fields. Then in a further experiment, the researchers put back human cryptochrome inside the flies, and the flies were back to being able to find food via the presence of a magnetic field. So all those things are consistent with the microscopic model. But then again, it's very hard, right? So what we're starting to do here at UCLA, we're super delayed because of COVID and stuff, but we persist. What we want to do is to bridge those length scales. What we want to do is, if you will, quantum sensing, chemically inspired experiments on cryptochromes in vivo inside, say, single cells. What we are building are different types of glorified microscopes with coils. Okay, so what we want is to explore those spins in proteins as bona fide quantum sensors that act at room temperature and in noisy environments, right? So if spins in proteins are treated as bona fide quantum sensors, there's this huge phase space for us to take technology from the field of quantum computing, quantum sensing, spin physics, atomic physics, and start applying those techniques onto those spins in biology. And I'll tell a little bit more about this, but before I want to mention that, again, this is not a talk about birds, okay? This is more widespread than that. Right now, there is, again, evidence at large length scales that this exact same physics of spin-dependent chemical reactions might underlie many, many relevant biosensing phenomena, such as metabolic regulation, stem cell mediated growth, in this case in planaria. There's also studies on human stem cell epigenetics that are altered by weak magnetic fields. DNA repair is altered by weak magnetic fields. This last picture that I post there, it has a story, right? And I'm going to spend my minus 2 minutes remaining to tell this story. So I have a friend in Munich. His name is Peter Fierlinger, and he's a precision measurement physicist. So for a living, he builds hypomagnetic chambers, and he puts cold atoms experiments inside to do precision measurements. And for that, they need very, very low spurious magnetic field. So he can build hypomagnetic chambers with DC magnetic field levels on the nanotesla range. Again, the magnetic field of the Earth is 50 microtesla. So just for kicks, what he did, and actually another group in Mainz replicated this experiment, is the following. They took tadpoles. Again, they're physicists, but they wanted to play with tadpoles. They took tadpoles and they grew inside those those hypomagnetic chambers under 2 different conditions for 2 days. In one condition, they grew tadpoles just with this DC nanotesla magnetic field. And in the other condition, they grew the tadpoles inside this hypomagnetic cage with an internal applied magnetic field mimicking the 50 microtesla magnetic field of the Earth. The tadpoles grown under 50 microtesla were okay. The tadpoles grown under the absence of magnetic fields were, as you can see there, about 30% macroscopically malformed and weird looking. Okay? This is not a proof that this is spin mediated, but given the tiny magnetic field strengths involved, my best educated guess right now is that this is spin physics uh, dependent. And again, this poses all sorts of questions, right? From disease sort of stuff, like can we start applying, start tapping on those endogenous quantum mechanical degrees of freedom to drive physiological processes in ways that we need to be able to predictably know, right? Questions from that to like space exploration, right? What's the magnetic field in Mars? Can we reproduce in Mars? Can we grow tomatoes in Mars, right? So I think that there's a lot of uh, interactions of biological materials with tiny magnetic fields via spin physics that needs to be explored. So this field is lies within a bigger field called quantum biology, right? And I maintain that right now the bottleneck for spin mediated quantum biology is the sort of high tech instrumentation that we are going after. When we say that we're building microscopes, the microscopes don't look like the microscope to the left of the pictures there. It looks like the thing to your right. It's like an atomic physics experiment with plenty of mirrors, electronics around that is run onto biological samples. I won't have time to talk about that, but it's very important that our experimental approach is sort of driven by mathematical models on this spin physics. What we want to do is model the spin physics and then make predictions and see where we are with this model in the lab. Even like we as scrappy experimentalists, we can already do um, simplified spin simulations on what's going on. The simplest model that one needs to use if you just do a open quantum systems Liouvillean evolution simulation is like you have two electron spins that start entangled, start in a singlet state, but that otherwise do not interact among themselves. Okay, this spin is free, and this guy has hyperfine interactions with, say, one nuclear spin nearby. With this three spin crappy simulation, you can already start making some very interesting predictions. And I promise that the last thing that I'm going to explain to you, but have a look at this top right plot here, where this is a simulation from our code. We're not the first people to do these simulations, but we can also do those simulations. So here we plot the magnetosensitivity of this putative of this cryptochrome using, again, this model plus photophysics parameters from the literature. So magnetosensitivity, that is how well the protein is at like distinguishing between the two pathways, the two chemical pathways. If the chemical reaction continues through 50% of the time through this pathway and 50% of the time through this pathway, this is a field for which there's very poor magnetosensitivity, right? Because it's almost random. But if for a given magnetic field intensity, frequency, or direction, the protein responds with the chemical reaction going 95% through this branch and 5% of the times through this branch, that's a very good magnetosensitivity. So our simulations is that the magnetosensitivity of this protein, right, as a function of applied external field, it's not monotonic, it goes up and down. At 10 times the magnetic field of the Earth, the magnetosensitivity is already very, very tiny. So going for stronger fields does not help, it harms, because again, this mechanism only works as long as those two electronic spins see different local magnetic field environments. If you start applying very strong magnetic fields, the hyperfine interactions that make all the difference are going to be overpowered by this external magnetic field, and changes in this external magnetic field are not going to cause differences in Larmor frequency phases. Okay, so this goes up and down. Interestingly, the maximum of the magnetosensitivity is very close to the magnetic field of the Earth, which is either a coincidence or it might show that those proteins evolved to work under the conditions where they evolved. And we think we can distinguish between those two hypotheses by doing, we cannot do this, but people who can do directed evolution of proteins could actually distinguish between those two properties. And that's the last thing I wanted to say for what we do. The mission of our group is to unambiguously determine whether quantum physics can be established to account for relevant biological phenomena, and importantly, be manipulated to technological or therapeutic advantage. Therapeutic advantage by driving physiology, right, in ways that haven't been mapped before. And technology, we also think that we can learn with nature how to build better quantum systems, especially because if nature is using quantum mechanics to function, it knows damn well how to deal with noise. Okay? We're not the only people actually thinking about this problem. There are other people doing high-tech experiments on biological systems, and I do think that interdisciplinarity is what's going to drive cool things in science forward in this century. So we've made the turn. I have to like take out my hat to the people in the group. I am so proud of these people who have persisted, starting a new group during a pandemic with a theme that is sometimes controversial. Those people are the reason why I wake up at 3 a.m. to give talks and to talk about our research. So that's all I wanted to talk about. May the quantum be with you, and I'm ready for more questions.
Okay, Clarice, thanks for a super talk. This was really cool. So much stuff here. I'm sure we'll have. Okay, so we already have a bit of questions. I see that we have questions from Alex, Lasek, Mercedes, and from Hugo. So maybe let's start with Alex and then go to Mercedes and then go to Hugo. Alex, let me just get you on the panel.
Hi, Clarice. I just wanted to ask you to elaborate on how these things you've mentioned, I guess both the color center and the Cryptochrome stuff, why do they remain, or why do they work while being quantum at room temperature exactly? Is it just because this microsecond coherence time is just long enough for the relevant process to happen?
Yes.
And
that's all there is to it. Those things wouldn't work. Neither Cryptochrome as a magnetosensor or the Envy Center as a magnetosensor. They don't work specially well or anything. Their working is not helped by quantum mechanics after the time that it thermalizes with the environment, right? The key to the thing is that all the sensing, the important sensing, is done before this thermalization process, right?
yeah.
And again, and that's why we want to keep it going for longer with pulses and decoupling sequences, right? But this is the whole deal. Quantum sensors at room temperatures, they exist. They're and like you can also, from a quantum sensing perspective, you can also make a sort of rule of thumb, right? The longer they remain coherent, the longer the coherence times, the tinier the difference in magnetic fields they can see, right? So quantum systems that decohere very fast, they can also be put to use as quantum sensors, but they can only sense big magnetic fields. The longer they're coherent for, the tinier magnetic fields they can sense, right? And yeah, and that's why they work.
Okay, yeah, I guess that makes sense. So biological systems, people are always skeptical that they could be quantum. So maybe they're not quantum in a quantum computing sense that it's a all-to-all entanglement, but they can sort of single-shot quantum experiments.
Nitrogen vacancy centers, to work as a quantum sensor, they don't need entanglement either. They just need to be coherent, right?
Yeah.
To work as a quantum sensor, a crappy quantum sensor, but a quantum sensor, you don't need anything else. You need coherence.
Yeah, I understand it now. Yeah, thanks.
And and another thing, like back of the envelope calculation is like, how sensitive magnetic fields can you sense, right? Again, there are better ways of calculating this precisely, but back of the envelope is like, you can sense a magnetic field if you are coherent for as long as it takes for you to do a complete Larmor precession, right? But again, back of the envelope calculation. So magnetic field of the Earth has a Larmor frequency of 1 point something megahertz. 1 over that is 700 nanoseconds. So in order to sense the magnetic field of the Earth, like bad limit, but you would need to be coherent for as long as 700 nanoseconds. And for the case of Cryptochrome, it's thought that it could be coherent for as long as 1 microsecond. So this is within reach. So
that's really cool. Yeah, thanks.
Yeah. Yes, it's super cool.
Okay, cool. Thanks, Alex, for asking some spot-on questions. Do we have Mercedes still on the call?
Yes, sorry, just one more quick question. Hi again, sorry. What is the advantage of the rotary echo pulse sequence over Floca engineering? I don't know if, I know it's like old, but yeah, so I was wondering if there's an advantage or yeah, when you were measuring, when you're using it to drive the, I forgot exactly what you were using it for, but you
well, I'm not sure if I'm missing some like specific thing that you're driving at Floca engineer, but this is sort of Floca engineer. It's like a driven quantum system, right?
so you're saying it's like the same.
I'm not sure. I might be missing the nuances of what you mean by Floca engineering. I think that in my crappy experimentalist view, Floca engineering is tied to designing sequences like driving periodic systems to do something. No.
Yeah, I mean, just to rephrase my question, then, why did you decide to use rotary pulse echo for the magnetic field manipulation?
Well, because this rotary echo can be tuned. The thing is a little bit what I said before. Usually the best sensitivity is gotten with a simple Ramsey pulse sequence. That is, you put your qubit in the equator, you let it evolve under the magnetic field, and then you put them back. And the phase picked up by the different spin states give you information on the magnetic field. In sensitivity, nothing beats this crappy sequence. Okay, but the sequence is very bad if your Z noise is very strong, right? Because when you put the qubit into the equator, it sort of dies out very, very fast if it's not driven. Okay, it sort of decoheres very, very fast. So this is one end of things. The other end of things is a continuous pulse, like a Rabi experiment. A driven quantum system lives for super, super long. Okay, so you can acquire your signal for much longer than you can acquire your signal for the Ramsey experiment. However, the sensitivity of your detuning goes as the detuning squared. That is, you're less sensitive to the detuning. And here for the Ramsey, it goes as the detuning to the first power. Rotary echo is something in between. Your sensitivity goes as the detuning to the first power, okay, but you still get some of the protection given by this continuous excitation, okay, by this continuous driving. This continuous kicking of the qubit helps it live for longer. And depending on how often you flip the page, you can actually show, I mean, there's a whole paper that I wrote on that. You can actually show that you can choose the flipping time to match the noise that you see in your system. That's the whole reason why we think rotary echo is cool from an experimental point of view.
And would you mind just going back to the slide where you're like, I think it's like 2 more slides back from this one? No, another one. Yes, this one. Okay, we're good. Sorry. No.
Yes.
I'm just gonna read that, and that's all my questions. Thank you.
Awesome.
Cool. Hugo.
Yes, hi. This was a really great talk, by the way. I really enjoyed it. So I had a question. You present these quantum sensors that then lead in biological systems that lead to macro behavior or macro expressions. So one...
Putatively, yes.
Right. One defining feature that leads to all the craziness of quantum mechanics and that puts quantum mechanics apart from a simple coin toss and a simple randomness is that you have these bell-like correlations that can persist over long distances, or that can have some correlations that are beyond normal probability. Would there be any evidence that this also happens in these biological systems?
So here's my best educated guess. I am almost, I personally, right, as a scientist, I have to do the experiment. But given the evidence, I do think that biological systems exhibit quantum superposition, coherence, spin coherence.
Mm-hmm.
I am yet to be convinced that there is entanglement in biological systems. Then again, it might be, right? It might be. I think it would be way trickier than actually what we already have to measure potential entanglement, right? And I think if we're still far away from unambiguously saying whether coherence exists, we're even further from knowing if there have been proposals of things in biology working because of long range entanglement and stuff. It might be, right? I am not personally convinced about that yet.
Okay, great. Thank you.
Okay, I think I know we're running out of time, but just a very quick, very general question, and kind of connects to what Hugo was talking about. If you had to like name and just guess, like unscientifically if you want, just like any guess, what would you say is the kind of biological area or the animal or the plant or the biological thing where you would expect the most quantum stuff to happen?
Many chromophores, okay, in redox reactions, okay. Yeah, and I think that, again, there's a lot of, it's really crazy because there are chromophores in cells that are not exposed to light, right? There's a lot of potentially photo-driven things in cells that are, say, in your liver, right? That do not see light. So I think that in redox reactions, flavoproteins in general, and I think this is super more widespread than thought. There are crazy papers from the 60s from Russian scientists. So Russian scientists are really cool because I think that Russia did not develop this stigma of magnetic fields, electric fields in biology that I think is present in the West. So I think those people continued with this research for a long time. So there's papers that say that phosphorylation processes are magnetic field dependent. There are like books on what has been tried and what has been seen by applying weak magnetic fields and what things change, and people see all sorts of weird changes in gene expression. I think it's super widespread. And again, this is not data driven. This is my best guess that movement of ions inside cells when transient, right? But that's fine because our quantum sensors are transient too. Movements of ions inside cells when they're viewed from very close by, those might be generating strong enough magnetic fields to shift this balance of like those chemical dependent spin dependent chemical reactions. So I think it's in many biological processes actually. Also spin transport, but I won't have time to talk about it.
Thanks so much, Clarice. Super interesting. Next time in the UK, please stop by, as Hugo said, for the lab tour. Obviously,
Yes.
you've been, but things has changed.
I
And for
hear so, yes.
and for the complimentary like post-talk dinner, and pint.
Awesome. Awesome. And you guys too, if any of you is in California, whenever we're free to roam about again, please let me know. We're always looking for grad students, postdoc, master students, people who want to spend some time with us. We're a cool bunch of people.